This chart categorizes health conditions. On the right, a disease requires an abnormal structural change. A disorder involves functional impairment without structural damage. A condition requires neither, while a syndrome is merely a collection of associated symptoms. Establishing these strict medical definitions is a necessary first step, because tracing the genetics behind them is rarely so straightforward. Complex genetic traits heavily blur the boundaries between functional disorders and structural diseases. While the vocabulary of medicine demands rigid classification, the genetic pathways driving an individual toward these clinical states are entirely fluid, relying on a vast network of interconnected variables. Consider familial breast cancer. It is one of the most thoroughly studied heritable threats in modern genetics, carrying a clear, observable pattern of risk across generations. Decades of research have successfully identified specific, high-profile genes associated with this familial risk, most notably BRCA1, BRCA2, and TP53. This diagram visualizes a statistical paradox. In the left pie chart, known genes make up the small red slice, 20% of familial breast cancer risk. The remaining 80% stems from polygenic predisposition, a vast web of unknown variants interacting with environmental factors. Crucially, these polygenic variants are largely latent. They sit dormant in the genome until they interact with specific environmental mutagens, like targeted radiation or chemical exposures, which activate the vulnerability and trigger the cancer. To accurately map heritable risk, research has moved from the identification of isolated mutations to calculating the combined impact of a polygenic matrix. Finding these hidden polygenic variants requires choosing the correct theoretical framework. The first is the common variant common disease model. This model assumes that the genetic markers underlying a disease arose only once, early in human history, before spreading through the population as common variations. Researchers locate these common variants using genome-wide association studies, or GWAS. This case control strategy works by directly comparing the haplotypes of affected patients against healthy controls to spot statistical differences in frequency. This approach yields results when the disease fits the historical model. In the case of asthma, GWAS successfully identified the ORMDL3/GSDMB region on chromosome 17, linking a common ancient variation to modern disease predisposition. However, the second framework, the rare variant effect model, deals with diseases driven by a multitude of recent, rare mutations. These are often de novo mutations that spontaneously appear in an individual's genome without being inherited from either parent. Genome-wide association studies fail entirely under these conditions because the variants are too new and too rare to show up in broad population comparisons. Researchers are forced to abandon GWAS and rely on extensive, localized sequencing to find the culprit. Schizophrenia perfectly illustrates this challenge. Routine population scans miss the genetic drivers because roughly 5% of schizophrenia cases are caused by these newly minted de novo mutations found only in the affected patients. Mapping a complex disease requires first understanding the evolutionary history of the target variants, ensuring the statistical model matches the mutation's origin. Once a genetic vulnerability is mapped, we have to track how it behaves in the real world. Many polygenic traits operate through a gene-environment cascade, where a localized physical breach sets off a systemic chain reaction. This flowchart tracks atopic disorders. Top left, genetic mutations in FLG and SPINK5 genes weaken the skin's epidermal barrier. The cascade begins when external factors, like the pH of common soaps or a bacterial infection, exploit that localized weakness, causing a complete breakdown of the skin barrier. With the barrier breached, allergens force a systemic shift. The immune system overreacts, creating an inflammatory environment. This progression is the atopic march. What begins as eczema systematically escalates, following the arrows down to the final box, eventually manifesting as asthma or allergic rhinitis. A severe respiratory disease can originate entirely from a localized dermatological defect colliding with an external allergen. Gene-environmental interactions also operate internally, where multiple distinct genetic pathways converge under the pressure of a single environmental stressor. Chronic obstructive pulmonary disease, or COPD, is the textbook example. This diagram maps the pathogenesis of smoking-induced COPD. While cigarette smoke is the obvious environmental trigger, the disease requires a highly specific matrix of genetic predispositions. We can isolate the first major genetic vulnerability here on the left, a deficiency in the AAT gene. Smoking naturally triggers an immune response, releasing proteases. Without a healthy AAT gene to inhibit them, these proteases destroy the lung matrix unchecked, resulting in emphysema. Panning down the chart, a completely different vulnerability is found near the HHIP gene on chromosome 4q31. The HHIP protein regulates the branching morphogenesis of the lungs. A mutation here permanently impairs the lung's inherent biological capacity to repair cellular damage after smoke exposure. The environmental trigger acts only as the ignition source. It is the individual combination of mutated internal pathways, whether an inability to regulate proteases or a failure in tissue repair, that physically constructs the disease. This exact dynamic, the strict interplay between genotype and environment, dictates susceptibility across nearly the entire spectrum of human health. This table catalogs specific examples of that interplay. Look at the APOE row. High dietary fat and cholesterol intake does not yield identical blood test results across a population. It triggers exaggerated serum cholesterol spikes, specifically in individuals carrying the APOE E4 allele. Moving up to the NA2 row, red meat intake correlates strongly with colorectal cancer in individuals carrying rapid acetylator SNPs. And right above that, low folic acid intake drives risk specifically for homozygotes carrying the MTHFR ALA222 Val polymorphism. Lifestyle choices and dietary exposures lack universal, identical effects. Their ultimate benefit, or their danger, is strictly gated by an individual's unique polygenic architecture. Researching complex traits now requires analyzing the combined effect of many variants, rather than searching for an individual mutation. Identifying a patient's exact level of risk requires applying the right statistical lens for their evolutionary history, deploying GWAS for ancient widespread variations, and relying on extensive sequencing to catch rare de novo mutations. Finding the gene is only half the equation. Accurate prediction demands identifying the specific environmental trigger that acts as the catalyst for the genetic defect. Human genetics functions as an active, environmentally responsive system.